Well, thank you for inviting me to speak today. It's a real pleasure to come and talk about our work that's been funded by Oracle Cancer Trust. It's a PhD Studentship, which actually just started this July. And the PhD student, Leah Palmer, is here in the audience, and she's got a poster she can talk to you about. And Ian McKenzie, who's also co-supervising this project, is also here. And the project is development of a molecular biomarker test for metastatic cancer stem cells and oral squamous cell carcinoma. So we have been doing basic scientific research into oral cancer for about 10 years now. Well, longer than 10 years, but particularly on this particular issue of metastasis for about 10 years. And what we want to do with this project that's been supported by the Oracle Cancer Trust is translate our basic research into something which can actually start to tackle some clinical problems. And well, the problem that we've been focused on here is that the standard therapy for oral cancer is surgical removal of the primary tumour, and then depending on the pathological indicators, there are then some clinical decisions that are made. And if it looks like the tumour has not metastasised, hasn't spread to the lymph nodes of the neck, then the tumour can be treated quite conservatively. Whereas if it looks like this tumour has spread to the neck, then often this what's called a neck dissection, where these lymph nodal regions of the neck, it's not actually showing up there, but these lymph nodal regions of the neck can be taken away in what's called a neck dissection. This has got a serious operation. We have not insubstantial impacts on quality of life, and so it needs to be very clear about have very good clinical indicators about who needs this surgery. The problem is at the moment, I've got these figures here. About 15% of clinically diagnosed positive patients turn out to be metastasis free. So that means some patients are getting this more aggressive surgery who actually maybe didn't need it and could benefit from more conservative treatment. And 25% of clinically diagnosed negative patients turn out to have nodal metastasis. So some people being treated conservatively who actually could have benefited from this surgery. So what we really want to do in this project is translate our understanding of oral cancer metastasis to generate a molecular biomarker test that predicts the likelihood of metastasis, and this would enable the aggressive treatment to be targeted to the highest risk patients. And there is a field of molecular prognostics. There are tests out there for other tumour types, particularly for breast cancer. There's about 8 different tests for breast cancer. One which I was highlighted here, which is one of the more successful ones, is EndoPredict, which is in clinical use in a lot of countries. And this is, I've quoted a German study here of resulting clinical decisions. 38% of patients had their therapeutic pathway changed as a result of this clinical test. 25% of patients were spared planned chemotherapy. So it had a big impact on clinical decision making, this test which predicted how likely the tumour was to metastasise and to spread. And this test was generated. What they did, they took a set of tumours, some which were known to metastasise and some which were known not to metastasise, and looked at the gene expression across the whole genome and the whole gene expression profiles of these tumours, and looked for those genes which are most highly upregulated in the metastatic tumours to create this signature of metastatic tumours. This was tried in oral cancer. It's been tried, but there was a lot more noise in the system in oral cancer. It didn't work out so well. And so what we want to do is use a similar method, but use our understanding now of the biology of how oral cancers metastasise to create a biologically driven prognostic signature to avoid to cut out that noise from the system. And oral cancer is actually an exceptional disease for this biologically driven method, because unlike some other tumour types like breast cancer, oral cancer is actually quite a consistent disease between tumours, at least in the HPV negative tumour type. It's quite consistent. We know there's a consistent mechanism drive metastasis between tumours, and so that means we've got something to aim at. And also, unlike some of the other tumour types like breast cancer, we've got very good experimental models in the lab for oral cancer. And so what we know about oral cancer metastasis, what we've been looking at for the last about 10 years now, this figure just kind of it summarises it in very basic terms, is that you have a whole lot of different cell types here, different identities of cells in the primary tumour, and some of these cells are minority cells, termed cancer stem cells, and these are the important cells that drive long-term tumour growth and progression. And we now know that these cells can undergo a process termed epithelial to mesenchymal transition, which you don't need to know the details of that, other than that it enables the cells to become migratory, and so they can actually spread. These dark blue diamonds we're looking at now spread away from the primary tumour, invade through the extracellular matrix, disseminate through the circulatory and lymphatic systems, and then seed tumours at secondary sites. So it's really these blue diamonds, these metastatic cancer stem cells, these cells which have undergone this epithelial to mesenchymal transition to take on these migratory abilities that we're interested in, to identify and characterise these cells and develop a signature for these cells, so that then patients will be tested to look for the presence of these cells, which then we predict give an indication of whether the tumour is likely to spread. And the model we're using in our lab, and the sort of basic research model to start the project, is this model of invasion in a 3D matrix. So we can create a 3D matrix in the lab which is mimics the environment of the tumour, and when we can use. Oh, that's great. Thank you very much. That's great. Thank you very much. When we use. So we have two different resources. We use cancer cell lines, which are oral cancers which have been plated out into plastic dishes in the lab, and these are a great resource because you can grow them forever and ever and get lots of cells. But to confirm what we're seeing, we actually get patient tumours straight out of the surgery, and this is actually a picture of one here, which has been broken down into small multicellular lumps, which are then seeded into these 3D matrices. And this is one here. We can see this little aggregate down the bottom here. And when you seed it into the matrix, the cells can invade out. So these ones going up towards the top of the picture with the red circle around them have invaded into the matrix. And what we've stained them for is two different, these are in the green and then the red. We've stained for two different proteins. One in the green, which marks these cells, which are the proliferative cells, which sit still and grow. And the red, so that's a protein called keratin, a pan-keratin marker. And then the red, the ones which are invading, are stained with vimentin, which is a marker for these cells which have undergone this epithelial to mesenchymal transition. And so actually now we've got this model where we can identify and characterise these invasive cells which are invading away from the tumour and start to develop a signature for them. And so the experimental approach we're going to use. And so I should add just that this actual picture here was an actual patient tumour that was taken direct from surgery. We also do it with cell lines as well, which grow in dishes. So the experimental approach is the objective is to use a cancer stem cell invasion model to develop an RNA signature for metastatic cancer stem cells. We can use this 3D invasion model I just showed you, where the cells are invading out of this aggregate into the matrix, and we can use cell lines and fresh tumour specimens. And then we're going to separate out the invasive cells from the non-invasive cells and characterise them based on. We've got some candidates from our previous work we can look at, but more importantly, we're going to do a genome-wide target screen. Using a technique called RNA sequencing, where you can look across the whole genome and look for those genes which are upregulated in these invasive metastatic cells compared to the ones which sit still, and to get a biomarker panel. And then we're going to do similar to what was done for the EndoPredict test in breast cancer, where we'll do a retrospective analysis of metastatic versus non-metastatic tumour specimens, so we can take RNA from metastatic specimens and non-metastatic specimens, and try and train the signature to identify the metastatic specimens. But rather than doing this sort of unsupervised method across the whole genome, we can now limit ourselves to these genes which are specifically upregulated in these invasive cells, hopefully thereby that way cutting out the noise. So just a little bit about our environment for those of you who don't know. So as I've already mentioned, with this project we aim to translate our basic discoveries, what we now know about oral cancer metastasis, into advances that can actually make a difference to patients. I'm really pleased that our Oracle Cancer Trust are supporting us in this work. We're in the right place. This is the Blizard Institute. It's actually an institute for translational research, and it's attached to the Royal London Hospital. So the big blue hospital, just down the road from here in East London, it's the Royal London, and we are. You can't see it over here, but it's a small orange sliver just next door to the Royal London Hospital. So we've got a really good clinical collaborations that provide us with the patient specimens and clinical data, so we can understand whether the patient specimens are metastatic and other clinical parameters. We've got really good facilities. This is an image that Leah, the PhD student, produced. She's already produced this from her first early work, and this is from a 3D automated screening microscope. We can actually make a 3D reconstruction of how the cells are invading in these gels, in these gel matrices. And actually, this is a video of a 3D reconstruction. Hopefully, so you can actually see again these same two markers. This green marker for the cells which sit still, and then you can see in red the cells which have actually started invading away. And then actually we've got software which can reconstruct this on an individual cell basis and show what the cells are doing. The work that we've been doing, our basic research up until this point, has been got a lot of attention. I think this idea of these metastatic cancer stem cells has got a lot of attention in the field. This is our first basic scientific paper back in 2011, and it's been cited by over 200 other studies. So I think there's a lot of interest in this method, and hopefully now we can translate it into something which is actually useful to patients. So with the support from Oral Cancer Trust, it's just an overview to show that we are going to use our understanding of how oral cancers metastasize and spread to develop a metastatic tests for metastatic cancer stem cells, which can be used in patient prognostics using this method of invading cells. And just to mention the next steps after we finish the study, there are obviously opportunities to apply for further funding, for example, from the NIHR, for a larger collaborative clinical study with our colleagues at Barts Health to perform a larger validation cohort. And there's also other opportunities from this model, particularly a personalised prediction of therapeutic response. Because we can grow individual patient specimens from surgery in this method, we have the opportunity here to test therapeutic compounds on an individual basis. And obviously the gene expression panel that comes out from these metastatic cells can also give us clues as to possible therapeutic targets, which we can actually use to try and then start to develop therapies for targeting these cells directly. So thank you very much. It's a pleasure to be here today and be funded by Oral Cancer Trust, and I'll be happy to take your questions if you have any.